all of these alleles, they all have the same copies, they have the same versions, they can mix and match tissues because they have the identical proteins, the identical cell surface receptors, they're exactly the same. If you're not identical, the vast majority of us are not identical, right, to other people walking around on the planet unless we have an identical twin, we are going to have variability at these alleles. This experiment is looking at how we figured that out. If you take a mouse, and you back cross it, crossbreed it, you do mother-son, father-daughter back crosses in the same line. If you do that 10 times for 10 generations, essentially what happens at the end of those generations, the progeny that you get out are genetically identical at their alleles, at their immunological alleles at least, actually at most of their alleles. They're essentially identical twins of each other, even though they may be different sexes, because we've bred out any variation by back-crossing them. Same genetic background against itself, against itself, against itself. You remove any heterogeneity, and you create an animal that has no variability in its alleles. Right? And we can do this in lots of different strains of mice to create mice that are identical so that we can do things like, if I know everybody's immune system is going to behave, how are they going to behave to different types of viruses? How are they going to behave to different types of bacteria? How are they going to behave being challenged by different types of antigens? Right? If everyone has the same background, then I know your immune system is going to behave the same. It's how does your immune system respond to those different pathogens? This can be really useful. But early on, we couldn't understand that there was this amount of variability in coding for all of these different receptors. And so it took people some time doing different genetic experiments to figure out how much variability there was and to truly determine how sensitive our immune systems were to slight variations. So this is just looking at these strains of mice, haplotypes. Haplotypes are regions of chromosomes, right? So we think about groups of genes that are located on the same arm, the same region of the chromosome, genes that generally move together, even when there are crossing over events. These are genes that are in the same regions as each other. They tend to stick together. So what are the genes that are located within these haplotypes? What variability exists on mice of these different backgrounds? This mouse strain, CBAJ black 6 or C57 black 6 mice are the typical little brown or black mice that you see in every lab photo ever. They have identical HLA alleles. And this makes it easy to do blood experiments, tissue experiments, all sorts of different types of experimentation in these mice because they essentially all have the same genetic immunological responses. So now I'm going to get at experimentation showing us how do we know that your alleles have to match or else there's tissue rejection. I've got a mouse that only has B alleles, and I've got a mouse that only has K alleles for encoding for their MHC receptors. I'm going to cross them. They're going to create, right, a progeny that has a mix of B alleles and K alleles. And we're going to ask, what is this animal? What types of information is this animal compatible with? So if we shift, this is just looking at skin transplantation. Who can I skin graft onto whom and have them accept each other's tissues? Got dad, mom. If I try to take an animal that is homozygous for their MHC alleles with B alleles and and I try to take that tissue and I put it on an animal that expresses only K alleles, there will be rejection. It's because their immune system sees those MHCs. It sees the antigen presentation molecules. It sees these things. And it says, these are not encoded the same as mine. These are different. I will attack and destroy those things, right? It's like if you put A or you put B blood inside me, my body would wig out. My body is like, no, antigens should not be on the receptor of red blood cells. They should be smooth. They should not have these proteins. This is not good. They got to go. It will kill and destroy them. Same thing goes for the tissue. Are these receptors on the surface? Are they exactly the same as mine, or are they different? They're not the same as mine. I'm going to reject. Now the cool thing, for heterozygote mouse, who's got a copy of some B alleles and some K alleles, they accept everybody's tissue because their immune system has learned to accept the B versions of MHCs and the K versions of MHCs because they're all going to exist on this animal at the same time. Right? So this is just playing the game, mixing and matching and see who accepts. Now here's the really kind of neat thing. I know that I can't give yellow mouse, right, orange mouse tissue. I can't give yellow mouse, right? I can't do that. They're too different from each other. But what if we take BK tissue and we try to put it back on a B or a K parent? Unfortunately, the B parent can see the B alleles and is fine with it, but can also see the K alleles and is not okay with it, will reject. Same thing goes here. K parent will accept K, but B is foreign, and it will reject that. Now, if you think about this simply, this is only two sets of alleles. So all you have to do is differ in half your alleles, and you're rejecting. Now think about the fact that you and I have hundreds of different alleles to encode for these receptors, right? It gets very, very complicated very, very quickly to make tissue matches. So we're not just talking about B and K. We're talking about scores of versions of each one of these and mixing and matching them together to have compatibility. The neat thing about our MHC alleles, we express them codominantly. So this is not like allelic exclusion when we build a BCR or a TCR, right? If I build a heavy chain from mom, I only build heavy chains from mom. Dad's heavy chains alleles are shut off. For your MHC alleles, all of your alleles are turned on. You make the versions that you inherited from dad, you make the versions that you inherit from mom. You codominantly express them on the surface of all of your tissue cells that are capable of expressing MHC1 and MHC2. So it's a lot of variability there. This just gives us some examples of numbers. So if I'm building a class 1 alpha receptor, how many different versions, how many different alleles exist of that class 1 receptor at each locus? Thousands at our A, B, and C alleles. Which ones do you have? I don't know. You got to get genotyped to figure it out. How many do you make of your class 2 alleles? Well, you got a lot more haplotype groups to pick from, but then lots of variation and versions in there too. Right? So there's a lot of variability that exists in all of us at our HLA alleles, both our class 1 and our class 2 alleles. And you are expressing versions that you inherited from your mom and your dad at the same time, right now. Yeah?
So those
haplotypes?
Yeah, those ones, are those how they do the genetic ancestry thing?
Yep. Yep, same thing. So what we do is we look at, we're going to look at a whole chromosome. you're going to look at a region of a chromosome that is essentially, the closer genes are together, the more likely it is that those genes, if they're being transferred or if they're being swapped in a crossing over event during making gametes, that those two genes come together. What we try to do is look at regions of chromosomes, areas where we know the genes are always linked together. So this gene and this gene are always next to each other and they always travel together. And so we look at these haplotypes to follow how those chunks of chromosomal information have traveled through generations over time. Neat stuff, right? We look at some of these genes because they are grouped in haplotypes. And it is easy to follow where some of these genes go over time. Now, the cool thing is we're all carrying, right, different versions. They can be mixed and matched, and you're expressing them all at the same time too. So this encoding for lots of different versions of these antigen binding receptors adds to the diversity of the antigens that you can bind and you can present to your T cells so that your T cells can then make the assessment. Are these antigens good? Are they neutral? Are they bad? And mount an immune response. There are 134 versions of DPB1. Where really are the differences in these 134 alleles, proteins? Where do we usually see the differences in the proteins? If we are looking at a class 1 receptor, almost none of the mutations are in the stalk. This is the stalk that anchors you into the membrane. Most of these bars, these represent point mutations or SNPs, areas where a different amino acid is encoded for in your genome versus your mom, or your genome versus your brother, your genome versus your grandpa. Where are these differences? These individual changes. The vast majority of the changes are found in the binding pocket. This changes how big is the pocket, how deep is the pocket, how wide is the pocket, what charge is the pocket, what can bind in the pocket. The more variability you have in the pocket, the more antigens you can bind. Variability is good. Really, really good for detecting foreign antigens, for detecting things that are possibly bad for you. On the opposite end, this is really bad. Diversity is really bad if you need a new kidney, if you need a new liver, if you need a new heart. Finding someone who has the same versions of alleles mixed and matched together is incredibly difficult. You may only match somewhere between 50 to 25% with your parent, and 25% are less with a sibling. So finding matches is really hard. Yes?
Identical twins would be perfect.
Identical twins should, in theory, be identical at all of their allele spots. There is a really weird phenomenon that can happen, though, and that is epigenetic markers can be different in identical twins, and sometimes that can cause immunologic responses that we would not expect to see in identical twins, but it can still happen. So, now that we've thought about this, what is in this second little PowerPoint that is called Visualizing MHC? It's a picture showing us. You've got all these alleles that are encoding for your MHC1s and your MHC2s. What does that mean? What would that look like? Look at all these versions of MHC class 1 receptors I can make. I make and express all of these. Look at all of these versions of class 2 receptors I can make. I make and express all of them. They potentially can be on the surface of all of my cells. So there's a lot of combinatorial diversity that can happen just mixing and matching protein pieces and parts together from all of the different alleles that you possess. So you make a whole bunch of different versions of class 1 and class 2 molecule that you are expressing on your tissues at any given time. This again is looking at some of the diversity, numbers of alleles. You can encode, there are 195 different versions of this A allele that exist out there in people, right? So what does that mean? Well, we got mom and dad, they've come together, these are their versions, these are the versions that we get out in their progeny, right? So we're gonna have blue and green, red and yellow, blue and yellow, green and red, lots of different combinations, mixing and matching, that will happen making gametes. Mom and dad's gametes come together and all of these different versions, right? New versions can result. This is showing us again polymorphism, lots of different versions of alleles, lots of different genes encoding for the receptors. Now we're mixing these two things together. There's lots of different genes and lots of different alleles coming together, and you get so many different versions of these receptors that can be made. It's all about diversity and antigen binding, making sure your cells can bind lots of antigens to show your immune system what's happening to you. This again is just looking at numbers, mixing, matching, all the different combinations that can occur and how new combinations are made. You might say, well, how did we get 195 different versions of allele A? Crossing over, point mutations, all sorts of different genetic anomalies can happen over time. And unless those mutations, those changes are selected against, so unless a progeny dies without passing on that genetic information, those alleles just keep coming along for the ride. They keep, right, expanding over time. Super neat. Okay. So, class 1 and class 2 receptors. Class 1 receptors engage with cytotoxic T cells. Class 2 receptors engage with helper T cells. Where do the antigens come from? Class 1 receptors are expressed on all of your nucleated cells in your body. Essentially, all the cells in your body, with the exception of red blood cells, express MHC class 1 receptors on their surface. The antigens that are displayed in those class 1 receptors come from inside the host cell. So I like to think about class 1 receptors are like a billboard on the surface of all of your tissues that are telling the outside world what is going on inside that cell. So right now, inside one of my liver cells, the liver cell is making proteins, doing translations, turning things over. I've got some ubiquitinated proteins. Those proteins are going to be chewed up. Some of those protein pieces and parts are going to be loaded onto a class 1 receptor. The class 1 receptor is going to be displayed on the surface of my liver cell. So that any immune cells crawling around in there, cytotoxic T cells crawling around inside there, can look at that antigen that's being presented by my liver cell and say, oh, that's just normal inside stuff. Okay, good job, liver cell, and keep crawling along. What if I am being attacked by hepatitis A? Hep A is a virus that is an internal, intracellular pathogen. It's inside my liver cells. It's growing, it's dividing, it's turning my liver cells into a factory for producing more viruses. Virus proteins are being made inside my cells, inside my liver cells. Some of those proteins get chewed up. They get displayed on a class 1 receptor. The class 1 receptor moves to the surface of my cell, and it's showing this Hep A antigen. Now Mr. Cytotoxic T cell crawls along and looks at the receptor and what is being displayed. It says That is not us. That is bad. That is a viral antigen, and the cytotoxic T cell will force my liver cell to undergo apoptosis so that it stops producing more viruses. Okay. So all of our nucleated body cells sample some of the proteins that are being made inside them and display them on their surface to show the outside world what's going on inside us, so that our immune system can know that inside the cells are healthy and happy, that everything is going right, Because how else would our body know? Oh, there's a virus inside there? I don't know. How would we know? It's hiding out in there. So we are capable, our cells are capable of showing, expressing how they are doing internally to the outside world. And it's class 1 receptors that do that. So on all nucleated cells. Class 2 receptors are only found on the surface of generally professional antigen presentation cells. A cell whose job it is to collect antigens and display them. So you might say, well, how do I know who's a professional antigen presentation cell? Any cell that does phagocytosis. Neutrophils, to a certain extent, macrophages, monocytes, dendritic cells, and believe it or not, B cells are also critically important professional antigen presentation cells. Like, that is their job. This is what I do. I crawl around, I collect antigens. That's my job. I'm a phagocytic cell. I collect antigens, I process those antigens, and I show them on a class 2 receptor. Those antigens come from outside cells, the extracellular world, right? So I'm a macrophage, and I'm squiggling around in between cells, I'm crawling, I'm in there, and I find a worm. Wow. Right? I start eating up worm proteins, worm parts. I digest them inside me. I load them onto a class 2 receptor, and I get up and I run around. I'm like, oh my God. I find a T helper cell, and I show the T helper cell, look at what I found. And the T helper cell says, oh my God, it's a worm. And starts releasing interleukin-4. Oh my God, it's a worm. Right? So that everybody comes. Everybody I need, all the other phagocytic cells, granulocytic cells, cytotoxic T cells, everybody shows up so that we can destroy that worm. So class 1 molecules come from inside the cell so that my insides can tell the outside world what's going on. Class 2 molecules are for the presentation of antigens that have been specifically collected by antigen presentation cells. Monocytes, macrophages, dendritic cells. B cells can also do this job, although technically their job is not really to do phagocytosis. They're wandering around covered in antigen receptors. If they bind antigen, they bring it in, they digest it, and they display it so that they can talk to T helper cells about what they found. So those are traditionally our professional APCs. Right, so examples. Professional antigen presentation cells, dendritic cells, macrophages, B cells, monocytes also fall into this line. Neutrophils. This is where life gets a little shady, right? Neutrophils are the jack of all trades cells. They do phagocytosis, right? They are the most highly phagocytic cell in our body. They also are granulocytes, so they can release cytotoxic chemicals into the environment. They can function as antigen presentation cells, but because they are the first line of defense, it's more important that they function in that first line. It's more important that they're eating, that they're releasing toxic chemicals, that they're working at that job, not that they're collecting antigens and then relaying them back to the boss. So neutrophils can act as professional APCs, but they usually don't. It's because they're abundant and they're busy, they have to do most of the work. It's the macrophages, it's the dendritic cells that specifically collect stuff, then bring it back to lymph nodes, to the spleen, to other places to show the antigens that they've collected. Here are some other examples of non-professional antigen presentation cells. These cells can often do antigen presentation through both class 1 and class 2 receptors. So here's an important thing for us to remember. All nucleated cells in our body have class 1 receptors and can show internal antigens. Is a macrophage a nucleated cell? Is a dendritic cell a nucleated cell? So all of your professional APCs have class 1 and class 2 receptors on their surface. This is really important for a phenomenon that I'm going to talk about in a little while that specifically dendritic cells do. But we know, right, that cytotoxic T cells engage with class 1 receptors and helper cells engage with class 2 receptors. If I am a dendritic cell, I can collect information and display it through a class 2 molecule. I am also a dendritic cell, so I eat stuff, I chew it up, I destroy it, it's come from inside me. I can display things on a class 1 molecule at the same time. Dendritic cells act as a bridge between helper cells and cytotoxic cells because they can talk to both groups of cells at the same time, relay information to both groups. Most cells cannot do this, or do not do this well. Macrophages and B cells don't do that well. So this is a really important role for dendritic cells. All professional APCs, though, express class 1 and class 2 at the same time. The rest of our body cells typically really only display class 1 molecules. There are some cells that are granted a little bit of license. They sometimes display extremely high levels of class 1 molecules as well as low-level class 2 receptor. Skin, primary barrier, right? One of the primary immunologic barriers. Your skin can be collecting antigens and express and display them through class 1 and class 2 receptors to help activate the right cells. Whole bunch of other groups of cells here that can also do this. Thymic cells, thyroid, endothelial cells, glial cells. Does everybody remember what glia are inside our brain? What's their job?
Structure and support.
Structure and support cells. They're kind of like brain macrophages. They can eat stuff in neurological tissue, break it down, and do antigen presentation. So the glia actually are probably one of the most important immunologic cell in our central nervous system anyway, because the vast majority of our B cells and our T cells are not